A warm welcome everyone. My name is Bharti Chauhan and I am currently working at IIMT College of Pharmacy, Greater Noida as assistant professor. So today I am going to deliver a lecture on steroids. These are the content of my lecture, which are introduction, nomenclature, stereochemistry and metabolism. So first I will discuss the introduction. Steroids are a group of structurally related compounds that are widely distributed in animals and plants. The term steroid applies to compound containing cyclopentane per hydrophenanthrene nucleus or skeleton. First we need to understand the term cyclopentane per hydrophenanthrene. Look at the structure below. This is a phenanthrene ring. All steroidal compounds are the derivative of phenanthrene. In this structure, you can see three rings are fused in angular mode. When we do the hydrogenation of the ring, the unsaturated carbon double bond will become saturated. Now, hydrogen will be attached on each carbon and forms per hydrophenanthrene. It means all the double bonds are replaced by addition of hydrogen and becomes saturated. Sorry for the inconvenience. So now when we add a cyclopentane ring at carbon 13 and carbon 14 position, then it forms cyclopentano per hydrophenanthrene. Next is steroids exhibit two principal biological functions as important component of cell membrane that alter membrane rigidity and signaling molecule. So how are steroids different from steroidal hormone? Steroid hormones that belong to the class of chemical compound known as steroids are secreted by three steroid glands, which are adrenal cortex, testes, and ovaries. All the steroidal hormones are derived from cholesterol. So, steroids are also made naturally in human body, and steroids are designed to act like these hormones to reduce the inflammation in our body. They are also known as corticosteroids, which we say cortisols and are different from anabolic steroid used by bodybuilders and athletes. So these are the important nuclei of steroids and I will discuss them one by one. First is guanine. This is the basic steroidal nucleus of the moiety, which contains 17 carbon atoms in its structure. Next, strain consists of 18 carbon and include estrogen. And strains are of three types, which are estriol, estrine, and estradiol. Carbon 19 nucleus is androgen, from which androgens like testosterone are made. Carbon 21 nucleus is of pregnane, which is used to form therapeutically important compounds like progesterone and corticosteroids. Carbon 24 steroid nucleus is of choline. It makes cholic acid and bile acid in the stomach. Next, last is cholesterol, makes cholesterol and other therapeutically active compound and have 27 carbons in its structure. Now, I will discuss about the nomenclature. Steroid consists of four rings, A, B, C, and D. You can see below this structure. A, B, C are six-member ring and D is a five-member ring. A per hydrophenanthrene ring ABC is completely saturated derivative of phenanthrene, while ring D is a five-member cyclopentane ring. Now, if we talk about the numbering, the basic skeleton contains 17 carbon atom and the numbering starts from ring A of phenanthrene, then followed by B to C, then ring D. First, let's start the numbering. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. I think you can easily see my cursor. I will move it and you will understand again. Now, let's focus on this structure. It is an example of a steroid which is cholesterol containing 27 carbon in its structure. Now, you can see the numbering that I have already showed you, which are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So that's how you can do the numbering of steroidal compound. Further, you can do the numbering here of the bond 18, 19, 20, 21, 22, 23, 24, 25, 26, and last 27. Next, If we talk about the configuration, you have to remember these two, alpha and beta configuration. You can see here, this is a solid line, which we say beta configuration. And below this, you can see the dotted line, which we say alpha configuration. Now, if we talk about the configuration, you can understand the moiety, which are present as below the plane or above the plane. Configuration, if we talk about the solid lines, the groups will always be present at above the plane. And if we talk about the dotted line, the groups will always be present at below the plane. If the configuration is not known, then it is shown as wavy lines. The configuration of hydrogen at carbon 5, I'm talking about this configuration which is dotted line, you can write it as 5 alpha androsten. This is an example of steroid. So if one double bond is present in steroid nucleus, the ane of the parent name is replaced by ene. And if two double bonds are then replaced by diene. If three double bonds are present in a structure, then it is named as triene. Okay, I have taken this example. You can understand by this. There is a double bond. Okay, so you can write this as ene. Here you can see. Next, the symbol delta is generally used to represent a carbon-carbon double bond in steroid nucleus. So you can see here, I've taken this example of 17-beta estradiol. There is three double bond are present. I have already told you that if there is three double bond in the structure, you will be named as, sorry, you can give this name as triene, which are at position first, third, fifth, and connected with 10. Why this is written as bracket 10? Now I will discuss about this also. If the carbon-carbon double bond is present between carbon 4, 5, it will always be represented as delta 4. At the starting, the double bond start with carbon 4. If the double bond started with the carbon 4, then it will be denoted as delta 4. And if carbon-carbon is present between carbon 5 and 10, that means if the double bond present in the carbon which are not in continuation, you can see here the carbon present between first and second, there is only one is written in this name. Next, uh, between 3 and 4, there is only written as 3. If we talk about the bond between 5 and 10, we will write as 5 bracket 10 because the numbering is not in continuation and the bond is not present in the continuation of carbon numbering. So I think you have already understand this. Next, you can also represent double bond simply by the number of carbon where double bond is present. If a steroid contains a 3-member ring, it is indicated by the prefix cyclo and the position number considered. If the 5-member ring is removed and we add the 3-member ring, then we will write the name as prefix cyclo. Next, compound with 5-alpha cholesterol are allo series and 5-beta cholesterol derivative are known as normal series. If there is any enlargement of the ring of steroid indicated by the prefix homo and preceded by a small capital letter indicating the ring affected, you can see here this structure. You can see my cursor, right? So this structure is at ring D. The ring is increased by the carbon atom. So this is not a 6-member ring. Now it is a 7-member ring. What we write here is where is the change occur at B ring. So we'll write B homo 5 alpha slash beta. Why 5 alpha slash beta? Because this is not, we don't know about this configuration. This is not dotted, not solid. So we will write it as wavy. We have shown here is wavy bond. So we'll write as 5 alpha slash beta pregnant. Okay. When a methyl group missing from the side chain, not prefix is used with the carbon number where methyl is missing. See, this is an example of androsten moiety. So here should be a methyl group. It is not present here. So we'll write what? 5 beta 19 nor androst. Here is not a methyl group. Methyl group is not present. So we'll write nor androst 3 on because of the presence of oxygen. Next, carbon number of substituent is placed in front of suffix. The carbon 13, carbon 10 methyl group are always in beta configuration. Next, I have taken these examples. You can now easily understand them one by one. I will discuss about them. I have taken first example as 17 beta estradiol. You can see here I have already discussed about this. Estra 135, 135 because 3 bonds are present. I have already told you 5 bracket 10 because the numbering is not in continuous and the double bond is not present with if the double bond is present in here. So I'll write only 5, but the double bond is present between 5 and 10. So we'll write 5 bracket 10 trines. 317. 317. What is present on 317 is hydroxyl group. So what we write here is it is present as beta configuration. So we'll write diol. 17 beta diol, and the name is made like that only. Next is testosterone. What is here? You can see 17 beta. 17 beta, it means it is beta configuration, solid line. So hydroxy, androst, 4-ene, 4-ene, 3 on. Next, preg, 4-ene, 3 on. Pregn, 4 double bond, ene, because 1 double bond, 3 on. It means 1 oxygen. Next, colest, 5-ene. 5-ene, 1 double bond, 3 alpha, all. Alpha, why alpha? Because it is a dotted line. Let's move on the stereochemistry of steroids. So stereochemistry of steroids is very important. Why it is important? Because stereochemistry affects biological activity of steroids. So it's more important to learn. There are two types of conformation, which is chair and boat. The chair conformation is more stable and have less steric hindrance. So all the cyclohexene ring are exist in chair conformation. You can easily find out the optical isomers also with the formula of 2 with power n. Here, the generic steroid structure drawn also has 7 chiral center, which means it may have as many as 128 isomer. How you can count? Let's see. The chiral carbon. What are the chiral carbon? It means all the groups which are present on carbon are different. So here, what you can see here, 5, 8, 9, 10, 13, 14 and 17. I have taken this example as you can see as I can discuss about the stereoisomer. But if we talk about the basic steroid nucleus, there is only 6 chiral carbons. So there will be 64 stereoisomers only. I have taken only the example so you can better understand this. ABC ring are in the chair conformation. It is the most stable form with least angular strain. I have already told you that chair conformation is more stable than boat conformation. That's why most of the steroidal compounds are present in chair conformation. Now we will talk about the cis or trans ring fusion. Two cyclohexane ring can be joined in either cis or trans conformation. Cis decalin. Decalin means two fused cyclohexane ring. Both angular groups at the ring junction are on the same side of the ring. It means you can see here what is the configuration? Beta configuration. Both of the groups are present at beta configuration. So we will say these are the cis conformation of these compound. It means A and B ring is present as cis conformation and B and C is at trans conformation. Why trans? Because one group is attached with alpha configuration and one group on beta configuration. It means one is above the plane and second is below the plane. It means it is present as trans conformation. Next, C and D group is present as in trans configuration also. So you can see one at the above the plane and second at the below the plane. Next, steroid may have either cis or transfused AB rings depending on the group in the fifth position. Other ring fused BC and CD are usually trans. So next, you can see this picture and I will tell you and discuss you about this more easily. You can see. So see this figure, you can see the hydrogen atom at carbon 5 has alpha configuration, which form the carbon 19 methyl group, which has beta configuration, and this opposite configuration make A and B ring junction trans. On the other hand, you can see the group are present as above the plane and have same configuration, so these are present as cis. Okay. Ring B and C fusion is trans in both conformation but only seen in natural steroids. This is point to be taken. The trans configuration between B and C are only be seen in natural steroids. Ring C and D fusion is trans and present in steroids, bile acid and related all steroids. So A and B ring have above the plane or group, so it is cis configuration. Next and the last topic of today's lecture is metabolism. In human body, the formation synthesis of different steroidal hormone is just the other way around from what we saw. We eat food, it makes glucose. Glucose makes acetyl coenzyme. Acetyl coenzyme goes into mevalonate pathway, which makes squalene, and from squalene, it makes cholesterol. And by cyclization, it forms other steroidal hormones in gonads or adrenal glands. Basically, steroids are inherently lipophilic molecules, and metabolic conversion are therefore required to increase their water solubility and enable efficient excretion in urine and bile acid. So, this metabolism is traditionally divided into two sequential stages, phase 1 and phase 2. Phase 1, all the reactions alter the biological activity and at the same time add or reveal functional group that function as target for subsequent phase 2 reactions. Now, what is happening in phase 2? Reactions are conjugation reactions that ultimately inactivate the compound and increase polarity and water solubility, thereby facilitating urinary and biliary excretion. And I want to add one more thing in this. Some steroids are directly undergo phase 2 metabolism without being subjected to a phase 1 reaction. Example, testosterone can be directly conjugated at its 17 beta hydroxy group. Now, steroid skeleton is synthesized from squalene. I've already told you this. The biosynthesis of steroid originate in cholesterol, C27 carbon, and include gradual breakdown of side chain. Pregnenolone is a metabolite of cholesterol. You can see here pregnenolone. So splitting of the part of side chain at C20 via enzyme P450 side chain cleavage monooxygenase. Next, it is converted to progesterone by conjugation. Next, it is converted to glucocorticoids, which is cortisol and also known as cyclocortisol. And progesterone also converted into mineralocorticoids, which we know aldosterone, with the help of enzyme hydroxylase and dehydrogenase. Lastly, progesterone is converted to testosterone and after aromatization, it is converted to estradiol. So hope you liked my lecture. Thank you so much for patiently listening to me.